Let's start new topic, that is cell biology, which is very important chapter and for the entrance course. And you should first know this, because this cell biology is the basic and fundamental to do all of the majority of the question, and used in all part of nearly in zoology also, in botany also. So let's get started. So first, study of cell is called what? Cytology. So cytology include what? Cytology include cell organelles and cell inclusion. So cell organelles is living part of cell. Cell inclusions is non-living part of cell. So in modern cytology, we should remember this. So in modern cytology include both cell inclusion and cell organelles, that is living part of cell and non-living part of cell, with along with physiology, means how cell done performance metabolically, chemistry of cell and genetics of cell. So mainly cytology deals with the structure and cell biology, structure and function of cell. So structure and cell biology, structure and function of cell. So cell is the structure and functional unit of life. This is a basic definition we learn from the childhood. Now Hartwig coined the term cytology, Mueller coined the term cytogenetics, Robert Hooke is known as father of cytology. Swanson is known as modern cytology, that is cell biology. Now Robert Hooke discovered. So first cell is discovered by Robert Hooke on the plant of bark, on the plant of outer bark of Quercus suber. So this plant is known as what? Oak. So oak tree you can say. So oak tree bark or Quercus suber. From this plant the cell wall is found, and that is cell wall is, we know that is dead and attached to cell. Now Robert Hooke published this about what? Cell wall of cell from the tree Quercus subericus or oak tree in the book Micrographia. Now Emil Leeuwenhoek is known as father of microscopy. He discovered living cell and free cell. Living cell and free cell, that is from the sperm. So sperm is what? Free cell and living cell, which is also moving. Now next is cell theory. So cell theory proposed by Schleiden. He is German botanist, and along with him Swan, who is German zoologist. Now in 1839 he stated that the smallest structure and functional unit of life is what? Cell. So according we studied cell. What is cell according to his definition. Now first idea cell theory given by Docherty on the plant Mimosa pudica. So Mimosa pudica is totally not plant. In nitric acid. So he put Mimosa pudica in nitric acid and separated this cell. Now exception of cell theory. So where the cell theory can't be applied. So cell theory can't be applied in virus, because virus is obligate parasite and subcellular. And subcellular means not acellular. It is subcellular, means not the form of cell but cell like. Now paramecium, which is binucleated. Cell contain only one nucleated, but here paramecium contain two nucleated, that is binucleated. Rhizopus and mucor in fungi, which is multinucleated. Vaucheria algae, which is multinucleated. Protist is also multinucleated. So a cell contains only one nucleus, but this organism don't follow the rules. So example you should remember something. Now cell theory extended by Rudolf Virchow. So Rudolf Virchow gave law of cell lineage. So cell lineage means how cell develop. So he told that cell originated from pre-existing cell and only by what? Reproduction. Only by reproduction the cell will come out. Now cell principle or modern modified cell theory is cell theory plus cell linkage. So this is cell linkage is also important. Now we have we have to know some of the basic and important point. So here smallest cell is mycoplasma. So only what? Only know this mycoplasma. So mycoplasma is smallest cell and it doesn't contain cell wall. So a study of mycoplasma, we should know that it's contain ssRNA, means rhino it's contain what? Rhino RNA, and also known as PPLO, means pleuropneumonia like organism. So it will perform pneumonia like disease, and without cell wall is called joker of plant kingdom, and some smallest cell with cell wall is bacteria. In animal longest cell is nerve cell. Example is sciatic nerve, which is from the hip to toe or leg. Now largest cell is ostrich egg cell. Now we have in plant longest cell is fiber of Rennie, and largest cell is acetibularia, which is uninucleated and belongs to green algae. Green algae is that algae which contain chlorophyll and chloroplast, and in this green algae a Spirogyra also comes. So here it is like what? Umbrella shaped. Now largest prokaryotic cell is Spirillum volutans. So this is also important. Largest prokaryotic cell is Spirillum. Now cell totipotency. Now cell totipotency is the capacity of living vegetative cell to regenerate into whole plant. Now this theory is first given by Howard Land, and the cell totipotential theory is experimented by whom? By Steve Ward. So Steve Ward told that in phloem tissue from the root of carrot uses explant, and this explant can form any type of cell. That is called what? Tissue culture. Now in tissue culture, which is what? Soma clonal variation. So tissue culture variation is called soma clonal variation. Now in tissue culture we have to use coconut milk as liquid media. So all this used by Steve Ward and his co-worker. Now coconut milk is like liquid media. So here coconut milk you perform function like cytokinesis, means power this cell division, means fast cell division occur. So coconut milk factor. Now he used what? He used plant virus free plant. So virus free plant is from a tissue culture. In tissue culture, from the tip of stem virus free plant is occur due to highly their division. So in highly division no cell, no virus or bacteria there survive. Now pluripotential is the animal cell potential to develop into animal body, but not whole animal body organ, you can say, or animal body organ. So in animal there is a stem cell which can perform this totipotential. Now cell ghost. So cell ghost is like when RBC or erythrocyte, when erythrocyte is passed in or kept in water, then erythrocyte chemical content will will what displace in water and out of the cell. That is called cell ghost. Means no content, no cytoplasm content there. So this is called erythrocytes or rupturing of erythrocytes or hemolysis. As we have studied in animal tissue. Now cell size. So normal cell size is up to 20 microns. So microns mu is 10 to the power minus 6. Now how microns? So microns used in light microscope 10 to the power minus 6, and angstrom is used in electron microscope. Now factor affecting cell size. So cell size, if cell size is increased, chromosome number also increase, size of chromosome also increase. If cell size increase, nucleocytoplasmic ratio decrease. So nucleocytoplasmic ratio is decrease, then cell metabolic activity also decrease. So surface area is also inversely proportional to cell size. So a small cell have a high metabolic activity. Now type of cell. Now we can define type of cell on the basis of nucleus, that is prokaryotics and eukaryotics. So in prokaryotics, nucleus is not fully organized, or it is known as incipient nucleus or primitive nucleus, where no covering is developed. Example is mycoplasma and cyanobacteria, or you can see bacteria. Now eukaryotic. So eukaryotic is what? Organized or true nucleus. Example is fungi, protist, plant and animal, which contain what? Well developed, organized, which contain covering. Next here is we have difference between prokaryotic cell and eukaryotic cell. So generally prokaryotic cell have no nucleus cover and eukaryotic cell have what? Nucleus cover. So let's get started. So here false nucleus, that is incipient nucleus or called prokaryons, which doesn't contain nuclear membrane, nucleoplasm and nucleolus. So these are the part of nucleus and that are absent here in prokaryotic cell, and here contain nucleus, that is called genophore or nucleoid. Now eukaryotic cell contain nucleus with nuclear membrane, nucleoplasm and nucleus. So these are what? Covers and main functions. Now here is in prokaryotic histone protein is absent. Instead of histone protein there is polyamide, which acts as a basic protein that neutralize the anionic charge of the DNA. Now in eukaryotic cell have what? Histone protein, and that is what? Basic protein. So histone protein is a basic protein which neutralize ionic charge of the DNA. Now in prokaryotic have only one envelope system and eukaryotic two envelope system. Prokaryotic intercellular compartment means intercellular compartment is absent, or membrane bound compartment is absent. Here membrane bound compartment means cover is present. Now in prokaryotic DNA is lies in cytoplasm and is double stranded circular. Here in prokaryotic circular DNA, which is non-repetitive and no histone protein type. Now in eukaryotic DNA is with histone protein with double stranded here and linear. So here is linear RNA, and in prokaryotic have what? Circular. So here is linear and repetitive means division with the help of histone protein. Now here prokaryotic DNA has single copy and here DNA has multiple copy. So according to DNA, so what is genome type is also selected. So from one gene one gene, one mRNA form. So it is what genome is single monogenomic, and here two type or main, that is polygenomic. Now here cell wall is non-cellulose and made up of peptidoglycan or murine, you can say, and lipid. Now here cell wall present is cellulose or chitin. So in eukaryotics we have cellulose or chitin present on cell wall. So chitin is like present in fungi, and genes are here continuous and genes are here discontinuous. Now linkage is group is only one and linkage group is two or more. So mainly linkage activate in eukaryotes. Now DNA RNA ratio is high in prokaryotic, and DNA RNA ratio is less in eukaryotics. Now ribosome only 70 type in prokaryotic and both 70 and 80 type in eukaryotic. So 70 type found in mitochondria, chloroplast, and 80 is found in nuclear membrane, RER, and cytoplasm. Now here microtubule and spindle fiber absent. Here microtubule spindle fiber present in eukaryotics. Now inhibition of transcription. So here the transcription is for conversion of transcription means what? Transcription means photocopy of DNA. So this is inhibited here by rifampicin, and here transcription is inhibited by alpha amanitin. Now here cilia and flagella is single stranded and made up of flagellin protein, and here cilia flagella is 11 standard. Here is only one single stranded, here is 11 standard. And so 9 plus 2 or 2 plus 9 arrangement, that is made up tubulin protein. So here is for flagellin and here tubulin. So here cilia and flagella combine to form tubulin protein. Now here aerobic respiration occurs in mesosomes in mitochondria, and here aerobic respiration in mitochondria. So here mesosome is instead of what? Mitochondria. We have alternative in prokaryotic is mesosome. Now here mRNA is monocystonic and polycystonic both, and here mRNA is monocystonic only. So monocystonic is like what? The division of RNA to form one gene to one mRNA. So here is only one gene to one mRNA, and here polycystonic has to be from many RNA to many mRNA form. So here cyclosis, mitosis, meiosis absent, and amitosis is common. Amitosis. Now here is cyclosis, meiosis present, and amitosis is rarely found. So in east amitosis is found, and here amitosis is commonly found. So this is difference. Now example is here bacteria, cyanobacteria, blue-green algae, archaea bacteria, mycoplasm, here rickettsia, etc. Many components of Monera family include, and here fungi, protists, euglena, plant and animal include. Now eukaryotes different from prokaryotes having what? Presence of cistern, because here mono and poly both cistern, but only monocistern in eukaryotes. Now here most excessively studied prokaryotes is E. coli. So E. coli is most studied prokaryotes. It is found in our intestine. Now chlamydia and rickettsia. So these are the animals which contain both RNA and DNA. So this is called connecting link between virus and bacteria. So rickettsia, remember rickettsia is a connecting link between virus and bacteria. Now here histone protein is positively charged, but what? But basic. You can say what? Basic. It is basic protein and is positively charged. So to neutralize the DNA. Now eukaryotic cell contain which are the following main type of DNA. So eukaryotic have linear and prokaryotic is circular type. Now prokaryotic genome differ from eukaryotic genome because it is. So prokaryotic genome has prokaryotic genome has no histone and it is circular and multi-stranded, so is non-repetitive. So non-repetitive is the best for what? Prokaryotic genome. Now we have next mesokaryotic cell. So mesokaryotic cell has nucleus with nuclear membrane but lack what? Histone protein. So lack histone protein in DNA, that is called what? Mesokaryotic. And cell division is here here is cell division okay by dimitosis. Now rest of the characters same to like eukaryotic, but best is what? Lack histone protein in DNA and nucleus in nucleolus in nucleus, but nuclear membrane is present. So like cover is present, but histone protein is absent in DNA and nucleolus is also. Now example is dinophysi. So dinophysi contain animals like dinoflagellates which contain lactiferase enzymes for the bioluminescence. So bioluminescence is that emitting it, so it is called what? Fire algae. Now eukaryotic cell means a denucleated cell, means no nucleus, that is called eukaryotic. So karyotic is basically nucleus. Now so that is example is RBC. We have studied in tissue, sieve tube cell. We will study in anatomy of plant. Now cell contains cell wall and protoplasm. So protoplasm contains plasma membrane and protoplasm. So protoplast, this is protoplast contain plasma membrane and protoplasm. Now protoplasm contain what? Protoplasm contain cytoplasm and nucleus. So cytoplasm contain organelle, inclusion and cytosol. So first is what? Organelle. So organelle is without membrane, without cover, is ribosome, centrioles, microfilament, microtubules. And that is microfilament and microtubules are called cytoskeleton. Now organelle bounded by single unit membrane. So single unit membrane is ER, Golgi body, vacuoles, microbodies, this type. Single membrane. Now with double membrane we have plastid mitochondria. Now with triple membrane is transosome. So T for T. You should remember this. This is the exceptional. Now nucleus is also a double membrane but not included under the organelles. And nucleus contain nucleus envelope, nucleoplasm, chromatin and nucleolus. Now inclusion means dead particles of cell, that is excretory is glucosides, tannins, gum resins. Secretory substances, coloring matter, enzymes, nectar. So reserved food is also present, that is carbohydrate, protein, fat, etc. And cytosol is also present. Now we have completed the charts. So from this chart we will study first cell wall, protoplasm, cytoplasm, organelles without membrane, with membrane, single membrane, with double membrane. So this type. So let's begin another class. Continuing here, we have three membrane organelle that is called transosome, and transosome is found in ovarian follicle in bird, in female for reproduction. Now when cytoplasm, from cytoplasm, when content of cytoplasm like cell organelle and cell inclusions are removed, that is called what? Cytoplasmic matrix, cytol, haloplasm or ground plasm. So here, when the cytoplasmic cell organelles and cell inclusions is what removed or make out from the cytoplasm, that is called cytoplasm matrix, cytosol, haloplasm or ground plasm. Now Golgi complex in or Golgi body in plant is known as dictyosome. Nucleus is absent in what? Prokaryotics, as we have discussed in prokaryotics and eukaryotic cells. Now cytoskeleton. So cytoskeleton comprises of microtubules or microfilament and intermediate filament. So you can remember by cytoskeleton is equal to MT, MF and F. What? MT, MF and what? And F. So this will be the what trick? MTMFF. MTMFF. So now endoskeleton. So endoskeleton means inside cell, that is called ER. Or you can say endoplasmic reticulum. Exoskeleton is outside cell, that is called what? Cell wall. Now cytoskeleton. So cytoskeleton, we know that cytoskeleton include MF, MT and F, or you can say MT, MF and F. So where it include MF, MT, and where is that F? So F is not given, so C should be answer. Okay, so C is the answer. Now here also we can say what? Network of microtubules and microfilament. Which of the following is considered endoskeleton? So endoskeleton here. Now cell wall is considered as exoskeleton. Plant cell different from animal cell. So what is present in plant and what is absent in plant, we should discuss. So plant contains cell wall. That plant contain what? Cell wall. Plant has also plastige and central vacuole that is large. So this is not found in animal. Also centrosome and centrioles with lysosomes are absent in plant. This is also for plant and this is also for plant. Now mitosis and meiosis is anestral in plant. This is in what? Plant. And astral in what? Astral or amphiastral in animals. So you can say amphiastral or astral is that spindle fiber. Spindle fiber is in contact with what? Centriole. That is called what? Astral. Now inorganic crystals common in cytoplasm. Sphygmosome, glycosomes present cytoskeleton by cell plate in animals. Cell is by what? Burying. This is all for what? This is all for lysosome. So lysosome is present in plant, is absent in plant but present in animal. This is all for animal. So animal contain cytoplasm, cytokinesis by what? Farrowing. We can study in cell division. And reserve food is starts in plant but glycogen in animal. Where plant cell doesn't burst in hypotonic solution, but RBC get burst in the solution. As we have studied one experiment done, and that is called what? That is dissolution of RBC. Now in plant all nucleated cells are totipotential, but in animal blastocyst or stem cell are what? Totipotential. Now we will continue to cell organization. That is first cell wall. Now cell wall common in plant, also in bacteria and cyanobacteria, and that is what? Non-living. So it is, so cell wall is made of non-living, contain plant bacteria, present in plant bacteria and cyanobacteria. Now its thickness is 10 micrometer up to, made up of what? Cellulose, hemicellulose and pectin. You can say cellulose, hemicellulose and what? Pectin. And cell wall is permeable. So permeable means it can withdraw and there will be soluble. That is called permeable. It is semi-transparent and covering secreted by what? Cytoplasm. Now in bacteria and cyanobacteria, cell wall is made up of murein, peptoglycan or mucopeptide, and peptidoglycan and polysaccharide contain NAG for chitin. Chitin that is present in fungi. This is present in fungi. This is present in what? Fungi. Here given fungi, chitin cellulose. So cellulose is also a type of glucose. Cellulose is the beta D glucose type, and peptidoglycan. So peptidoglycan contain in bacteria. So this is for bacteria, peptidoglycan for bacteria that contain what? NAG and NAM, and NAG is also contained in fungi. Now next is plant cell. Now plant cell without cell wall is what? Mycoplasm, slime mold, zoospore. So this is, now cell wall during cytokinesis from, so cell wall formed by what? Golgi body vesicles and ER. So main is what? Golgi body. Golgi body is main for forming the cell wall on the process of cytokinesis. Now cell without cell wall is called protoplast. We have studied in the chat. Now in higher plant cell wall different layer, that is what? Mainly 4 type of layer: middle lamella, primary cell wall, secondary cell wall and tertiary cell wall. Now first go to primary cell wall, contain outer layer, and this is adjacent between two middle lamella. So you can say 2 cell wall, that in between 2 cell wall that contain middle lamella, connecting layer to cell. Now it is made of what? Calcium pectate, calcium magnate, mainly calcium pectate. As you can say, calcium is helps to make what? Bone. So you can remember. Now when ripen fruit occur, then that soft due to what? Dissolution of middle lamella. So due to middle lamella softening, fruit become what? Ripe, and dissolution by pectinase enzymes. Now first layer to be formed is what? By Golgi bodies and ER. Now during cytokinesis, vesicles of Golgi bodies and spindle protein come in contact, called as what? Phragmoplast. So phragmoplast is the process of making. So this is what? This is important. Phragmoplast is the process of making what? Cell wall. Now phragmoplast from cell plate. So from cell plate in plant cell, and from the burrow fusion is by burrowing by animal, but animal doesn't contain cell wall. cell plate during cytokinesis. Middle lamella totally absent in unicellular plant and all fungi. So in unicellular plant and all fungi it is absent. Now we have what? Primary cell wall. So primary cell wall is elastic in nature and it is found in all, universal. This is what? Universal, found in all. Inner two middle lamella made of cellulose, beta D glucose, hemicellulose and pectin, like same that was before we have talked. Now starch is formed by what? Alpha D glucose. Now lignin is never found in primary cell wall. Global insertion. So it is mainly present in this primary cell wall, mainly present in parenchyma and meristematic cells. Now secondary cell wall, this is what? Thick and inelastic. It is inner to primary wall. It is triparticle, and where S2 is the thickest layer developed by apposition, means deposition. Pits is here common. So pits is the pore. From that it can exchange, provide mechanical strength, and additionally cellulose. It is made of what? Cellulose. So cellulose is made in all. Now here it contains additionally compound, that is cutin, which is what? Waxy substance, and from cutin there from what? Cuticle. If that cutin is not reduced and continuing from epidermis, then cuticle form, and that cuticle can be lost due to loss of water. Now cuticle is thick in xerophytes, thin in mesophytes, and absent in hydrophytes. Now we have second, suberin. So suberin is in cork shell and endodermis. Suberin is in cork shell and endodermis. So this is cork shell and endodermis. We will study in animal anatomy, plant anatomy. So checks for evaporation of water. It is mixture of fatty acid. Now pectin is in collenchyma. You can remember by cope or peco, pico, PECO, pico will be the trick. Now cellulose. So cellulose is in flax and cotton fiber. The most abundant carbohydrate is cellulose, and this insoluble also. Now lignin is found in xylem vessel, sclerenchyma, and in trachea too. Now lignin contain non-degradable methanogens during biogas formation, and it gives higher rigidity, means it is highly what? Strength and highly rough, and which lignin is also permeable to water. You should remember this. Now we have wax. So wax is on floating leaves silica. So silica is contained on the cell wall, that gives in the rough touch on the moraceae, cucurbits, calcium oxalate, raphaceae, calcium carbonate. This is type. Now mucilage is complex carbohydrates. Silimi, it is slippery. So plant got slippery due to mucilage, and due to this mucilage enzymes, plant can withstand in extreme temperature. Example, aloe vera, China rose. Now tertiary wall. So tertiary wall contain purely cellulosic in nature. So tertiary wall is what? Contain cellulose, purely cellulose. So all contain cellulose. You should remember the trick. Now it is mainly contain what? Xylem, and thick and warty like appearance. Example of its occurrence is trachyid of gymnosperm. So trachyid of gymnosperm. The phenomenon of formation of primary. So forming primary cell is called insertion, means consist of cellulose filling. Now for forming secondary cell wall and tertiary, that is opposition, means deposition on the layer Now plasmodesmata are living cytoplasmic, that is connection between two cell or adjacent cell. Now through plasmodesmata, protoplasm make what, make continuing system, that is called symplasm, means living system for what, connection. Now evoplasm is non-living system. Plasmodesmata is modification in cell membrane that is found in multicellular plant. Now unthickened area, so where the where not thickened, that is called what, pits. Now dead cell are interconnected by means of pits. So dead cell is connected by pits, because inside pits there is what, connector. So now pits are found only in secondary wall. So secondary wall is for the pits. Now pits contain also cavity and pits membrane. So we have studied that. So like this is like cell, and this is the space, and where all this is like what, this is rigid and non-permeable, but this is what, unthickened, and their substance can be passed. Now type of pit, that is simple pits is uniform in diameter, found in angiosperm, example companion cell and sieve tube, that is of phloem, phloem, phloem. Now bordered pits, so bordered pits is in cavity, contain pits chamber present in mainly gymnosperm, bordered pits. And torus, so torus is what, swollen portion, act as what, valve in vessel and trachea. Now sclerenchyma fiber has simple pits and border pits both. So sclerenchyma is what, our mechanical type of tissue, so contains simple pits and border pits. Now aspirated pits, that is non-functional pits, and predators by torus. So torus is mainly swollen portion of pits and which act as valve. So torus is like valve. So torus is always valve. Cell wall formed during what, cytokinesis, mainly by Golgi body. We have two, that is Golgi body and ER, but main is what, Golgi body vesicles is main. Now three vesicles fuse with spindle fiber. Now these vesicle, Golgi body vesicles fuse with what, spindle fiber to form phragmoplast. So phragmoplast is the cell plate. So phragmoplast is the what, cell plate. So when the vesicles with spindle fiber in contact to make cell plate, that is called phragmoplast, which grow also centrifugally, means away from the center. Animal cell wall is what, absent, and outermost covering, so outermost covering, alternative given outermost covering, that is glycocalyx or cell coat we can say. Now cell coat or glycocalyx is made of oligosaccharide. This is also a type of what, salic acid, or you can say oligosaccharide is a type of carbohydrate. Now some type of point here given. So cell wall is permeable membrane, exception is cork film, that is impermeable due to what, suberin. So cork and film is impermeable due to deposition of suberin. Now cellulose is most abundant. Cotton contains 90% highest amount cellulose. Chitin is second most. So chitin is what, second most carbohydrate. So here is that if beta D glucose, that is cellulose. So this is like, now structure unit of cell wall contain micelle, microfibril and macrofibril. So young meristemic parenchymatous tissue is the thinnest and xylem vessel is the thickest cell wall. Now matrix contain what, cellulose, but hemicellulose is mainly in main carbohydrates in matrix. Cellulose, hemicellulose you can say, but mainly is what hemicellulose that is given. Now cell membrane or plasma membrane, that is coined or term given by Kramer and Nageli. So you should remember this, Nageli and Kramer. Now Singal and Nicholson term biomembrane. Robertson gave term unit membrane for the cell membrane, plasma membrane or plasma lemma. Now Pfeffler gave plasma membrane, Plover gave plasma lemma, Plover. So P for P, you should remember P for P and P for P also. Now cell membrane is what? Thin, selectively permeable, and made of what? Phospholipid, made of phospholipid and protein. Now phospholipid contain head and tail. So phospholipid is where head is hydrophilic, means towards polar towards what? Accepting water, and non-polar means hydrophobic, means reject water. So here DNA is present but RNA is here what? Absent. So DNA is present but RNA is absent in plasma membrane. So it is like bilayered, and phospholipid is amphipathic, as we have know that is both hydrophobic and hydrophilic nature. So it is as amphipathic. Now plasma membrane plus organelle membrane is called biomembrane. Now lipid is more than protein here, but where lipid is more than protein, but previous it was told what? Protein should be more than lipid in the cell membrane. But this is only case of what? Myelin sheath. It is what? Elastically also depend upon the lipid. So lipid depends upon the lipid. Now thickness is 75 angstrom. Now forms of lipid, that is phospholipid, glycolipid and cholesterol. Now forms of protein, so it contains protein also, that is extrinsic means outer protein 25%, and inner protein, intrinsic protein, that is 75%. So mainly cell membrane is what? Permeable or selectively permeable, that contain more protein than lipid. So phospholipid structure. Forms of carbohydrate, that is glycolipid and glycoprotein. So different models and theories given for what? Plasma lemma, plasma membrane, or you can say what? Cell membrane. So first, that is, so generally it contains what? Lipid and protein, more protein and then less lipid. But actually in middle lamella there will be more lipid. So according to lipid, this is derived. So first, that is lipid model, that is proposed by Overton, suggests that plasma membrane contain of only lipid membrane in the cell but no protein. Now trilamellar or sandwich model. So sandwich model given by D, or you can say Daniel and Davidson. According to them, outer should be protein, middle should be lipid, and inner should be also protein. So like what? Like sandwich. In sandwich, between part is the main, that is what? Vegetable and like some things like that. And outer is like what? That is bread. So like this, outer and inner both are protein, and in middle there should be what? Bilayered phospholipid. And you should always remember what? Plasma membrane is always bilayered. Now total thickness is 75 angstrom. Now how is this 75 angstrom given? So 40, and there should be what? 25, I think. Okay, 25. No, there should be what? 35 and 40. Okay, this is here 35 and 40. Okay, so 35 and 40. So 35 is for lipid and 40 is for protein. So more protein. So this is for 44 protein. Now protein is like globular type. So protein can be much more type like globular and fibril. Now unit membrane, that is going by Robertson. Now according to this, protein was fibril type. So I have told you protein is of fibril type. So here plasma membrane. So his sequence was like this, same outer protein and between bilayer is, this is what bilayer lipid. Now greater membrane model proposed by Lehinger, which say, in which he says oligosaccharide present on what outer surface of cell membrane. Now fluid mosaic model proposed by Singer and Nicholson. This is most accepted model and based on freeze etching or fracture technique. Now this model differs from the both unit model and trigeminal model, that protein is on iceberg of sea of lipids. So like whole protoplasm is like whole cell membrane is like lipid, and there in between where protein lies. So like between is the protein. So between is the protein. So like this. So protein molecules are randomly and lipid are continuous layer. So like this. Now protein is here 25%, extrinsic, intrinsic 75%, and tunnel, that is in between. Now protein is here also globular type. He follow that past. Now he says lipid and protein. So lipid and protein are together by hydrophobic attraction. Hydrophobic means non-watery condition. Now rotational movement is like flip-flop mechanism. Rotational movement is flip-flop mechanism. Now extrinsic protein contain ATP and spectrin. So spectrin gives shape to RBC. Now intrinsic protein contain cytochrome, permease and rhodopsin. So extrinsic contain ATP and spectrin, and intrinsic contain cytochrome, permease and rhodopsin. So a special point, all carrier protein are intrinsic protein. Now also contain what carbohydrates on the outer surface. Now carbohydrates in there should be what form of lipid and glycoprotein or glycolipid. Now contain this, both is what site of recognition. So glycoprotein and glycolipids are sites of recognition. So glycocalyx made of this both, glycoprotein, glycolipids. Now glycocalyx is cell membrane asymmetrical. So layer of carbohydrate is oligosaccharide in animal and help in cell recognition. Now function of plasma membrane. Control what? Control the passage of material in and out by the help of calcium and camp. So here also exchange of gas CO2 and O2 through the membrane. So it is also act as what transport, here also transport by the help of what calcium and camp. Now transmembrane transport. So transmembrane transport, that here it should be permeable type and bulk transport. So in permeable transport, that should be active and passive. So active in bulk transport and passive or active here in osmosis diffusion and facultative diffusion. Now bulk transport, so from this bulk transport is here, that is active, that contain endocytosis and exocytosis. Endocytosis is mainly for the mainly for the intake, and exocytosis is the vomiting or making out. Now endocytosis mistaking in solid and intake of liquid. So solid called as phagocytosis and pinocytosis is for liquid, and also that is called cell eating phagocytosis, and pinocytosis is called cell drinking, that is liquid, for liquid that is drinking. Now exocytosis is for reverse phagocytosis and reverse pinocytosis. Now some point. So point is like, so active transport is by carrier protein, which require what, energy, and passive transport is neither protein or nor 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 energy required. So facultative diffusion doesn't require energy but require what, carrier protein. So this is like exception. Now modification of cell membrane. So from the cell membrane, we can form what, mesosome. So mesosome is the infolding in bacteria that helps in respiration and sputum formation. Respiration is sputum formation. Now lomasomes, that will be what, outfolding in what, lomasomes is present in cell organelles like fungi. So it is outfolding of cell membrane. Now here is lamosome. So lamosome is infolding of cell membrane in blue green algae, that is expansion of cell wall. Now Johnson complex. So Johnson complex is multicellular adhering. So Johnson complex is for what, joining or adhering, cell adhering technique like desmosomes, tight junction, gap junction, interdigitation, and terminal bar. So like microvilli. So microvilli is like finger like for digestive tract absorption of liquid. Now here given some question, that is cell membrane. Who are flowing cell membrane helping cell recognition. So cell recognition contain what, glycolipid and glycoprotein. So option C. So option C is answer. Now here also we can see D, that is oligosaccharide. So both contain, both is this contain is what, oligosaccharide. So best option is oligosaccharide. Now this is what, protocytoplasm. So this is like protoplasm. So protoplasm is the living matter of cell, first observed by Corti. So Duraidin discovered. Now Pukonjiki coined term protoplasm. Now von Mohl called the fundamental unit of life. So Max Mischle protoplasmic theory, that living matters contain protoplasm. Huxley say physical basis of life in all process. A protoplasm is polyphasic by Collier. Now protoplasm is biochemical, and protoplasm means what, cytoplasm plus nucleus, we have told. So cytoplasm contain that two, cell inclusion and cell organelles. Now best material to study protoplasm is slime mold because of absence of cell wall. Now protoplasm is highly active and with maximum energy conversion. Now living beings different from non-living due to what, protoplasm. So it contains mainly 80% of water and protein, with water 80%, we have told 80% what, water, and consist element. Now 62% oxygen, 10% hydrogen, 20% carbon, nitrogen 3%, that is low. So ratio in organic, that is organic matter contained in this protoplasm is 9. And protoplasm exists in two forms, that is soul and gel. So soul is like inner and gel is for outer. So this is opposite, like O for outer, so not, this is opposite. So gel is like jelly type. Sol gel, so sol gel interconversion is the what colloidal nature of protoplasm, because it is made of two forms, that is, or it is in existing two form, that is sol and gel. So outer gel is ectoplast or plasma gel, and inner, that is called ectoplast. And conversion of plasma sol into plasma gel is physiochemical phenomenon. Also cyclosis occur in endoplast region, means in inner part of, that is plasma wall, plasma sol, inner, that is plasma sol. Now Brownian movement or Tyndall effect and cyclosis, this all are colloidal in nature of protoplasm. So mainly colloidal in nature in the cell body. Now protoplasm coagulate in above 60 degree, which is treated with concentrated acid or base. So enzyme degenerated at high temperature. Now cytoplasm contain matrix, cell organelles and cell inclusion. So cytoplasm include these three: matrix, cell organelles and cell inclusion. In cytoplasm is cytoplasm, all structures are called haloplasm. Now ribosome contain what? Ergastoplasm, ribosome. Now deutoplasm is non-living substance. Now other point is like largest component in cell component is nucleus. Largest cell organelle in plant is chloroplast. Largest cell organelle in animal is mitochondria. Second largest organelle in plant is mitochondria. In animal, that is first. In plant, that is second. In animal largest is what? Mitochondria. And in plant, that is second largest. So in plant, which is biggest? So biggest is chloroplast or plastid, you can say. Now smallest cell organelles is ribosome, because without membrane. Now colloidal theory of protoplasm, that is given by Fishery and Hardy. They propose that in the colloidal system. Now cyclosis means a streaming movement shown by what? Protoplast. Now rotation is like in only one direction, that is clockwise or anti-clockwise around vacuoles. Now circulation is like moving in different direction inside cell, that is example like tradestendia. Now response to protoplasm to the stimulus is called irritability. Specific gravity is greater than 1. Now pH of plant protoplasm, not in different time, but generally pH is like neutral, which is considered at 7. So in animal, nucleus is alkaline, that is 7.6, and in cytoplasm is like acidic. 6.9, 6.8. If injury decrease pH, so when injury occurs, so decrease pH, that could be up to 5.5 pH or 5.2, and remain for long time in the cell and the cell type. Now cytoplasmic organelles. So cytoplasmic organelles, we have a mitochondria here. So double membrane structure contain mitochondria, plastid and nucleus. So in plastic chloroplast come. Now first is mitochondria, which is called what? Powerhouse of the cell or ATP mill, that makes what? That makes food for the plant by the or in plant or animals by the process of respiration. Discovered collicles is main important on the striated muscles of insect. Now size ranges 1 micrometer. Now Benda use term mitochondria. Altman give term bioplast. So bioplast is also for mitochondria. Now this is absent in prokaryotes, anaerobic eukaryotes, present in all aerobic and aerobic eukaryotes except mammalian RBC, sieve tube phylum. Now RBC at maturity obtain energy by glycolysis. So this is also important. So RBC receive energy from the what? Glycolysis process. Now mitochondria is double-walled, DNA contain and self-replicating. So it is known as what? Semi-autonomous, because ability to divide due to DNA and synthesis of protein, also contain RNA plus ribosome. So DNA is here what? Simple, circular and double stranded, which is 70s type, and endosymbiotic or prokaryotic like nature, means prokaryotic cell like nature, due to this DNA type, which is simple, circular and ribosome type, that is 70 years, or cell within cell you can say. Now chloroplast is semi-autonomous also and endosymbiotic. So autonomous structure means fully autonomous, means contain all that is nucleus only. So you should remember that autonomous structure is nucleus only. Continuing our mitochondria. So here minimum number of mitochondria. If one cell contain only one mitochondria, so that in plant cell, in cholera and microstatus. Now in animal we have trypanosoma. So trypanosoma we have studied that is a sleeping sickness. So it induce what? Sleeping, called Trypanosoma cruzi and Trypanosoma giardia. Now mitochondria is smallest in yeast and largest in oocyte of Rana pipiens. So Rana pipiens is the type of what? Amphibian. Now mitochondria second largest in plant but largest in animal cell, and third largest after nucleus and chloroplast. So in plant it is first nucleus is largest, then second chloroplast is largest, then third is that mitochondria. So here is second organelles, because nucleus is not containing organelles, it is separated. Now inner membrane enfolding, that is cristae. That is what? That is called cristae, or microvilli in plant. In animal that is called cristae, or no, in cristae in animal and microvilli you can both say. And cristae is site for what? Oxidation reduction due to presence of their what? Porins. So porins is here present, that is permeable porins, which act as channel for the entry and exit of what? Protein, where inner membrane is impermeable and this outer membrane is permeable due to porins. Now inner membrane bears ETS, that is electron transport chain, and tennis like, that is called oxysomes. Like this is like mitochondria, 2 layer, and it contain like this type cristae. Now in this cristae, contain like this tennis like this structure, where this is called what? This is called oxysome. So this oxysome contain differentiated into 3 parts. So these 3 parts called head, stalk and base. So here we will go through it. Now outer mitochondria contain what? Adenylate kinase enzymes, and oxysome differentiate head, which is called what? F1 particles, and base, that is called what? F0 particles. Base is what? F0 particles, where F1 particle is for head. F1 particle what? ATP synthesis, and this is the what? Final step for ATP synthesis in the head. Now stalk is for what? Conduction, second, and third part is what? F0 part, base, which contain ETS system, electron transfer system, and has what proton channel also. So proton what transport is here occurs. Now previous it was saying that two proton pass for one ATP. Now again, now in modern we have what three electron passed for one ATP in modern theory. Now oxyosome is called what functional unit of mitochondria. So how oxyosome happen? So like in inner there is Christie. So Christie contain tennis like or destructive departure structure that contain oxysomes. So oxysomes is that occur. Now 70% of total enzymes found in mitochondria here enzymes like Krebs cycle which is found in matrix and all enzymes contain in matrix except succinase hydrogenase which contain in inner membrane of mitochondria. Now we have what here note is that for fatty acid enzymes all present in matrix except ATP and cytochrome oxidase that is on the inner that contain on the inner portion. Now ATP production or synthesis in the mitochondria is called oxidative phosphorylation, and matrix of mitochondria contain Am and manganese. So dye for mitochondria staining is Janus green. Also mitochondria in a cell called chondriosome. So mitochondria different name chondriosome, ATP meal, mitochondria. Now mitochondria outer membrane removed that is called mitoplast. Lifespan is up to 10 days or you can say 1 week. Now peptide character in yeast is mitochondrial inheritance. No Christie in sperm. Now biogenesis mitochondria divide by binary fusion to form new one due to DNA that help in formation of middle piece fatty acid. So fatty acid two enzyme that is ATPase and cytochrome oxidase that doesn't contain on the matrix that is contained on the inner side of matrix. So now we have next plastic. So plastic discovered by and given by Haeckel's largest cell organelles in the plant because it is not found on the animals, mainly it is found in plant. And curtain protista too, in protista family too there is like dinoflagellate diatoms that types of that contain what plastic, and it is absent in fungi, double walled and DNA contain cell organelles. Now for plastic origin from pre-plastic that is called proplastic or eoplastic of the division by meristematic. Now plastic carries genetic information that is called plastidome. Now involved in storage of carbohydrates. Is the for main plastic is for storage of carbohydrates function and formation of what is formation of ATP, no formation of what food. Now this contain three types formation and what involved in formation and storage of carbohydrate that is like food. Now three type on the basis of pigment. So first is leucoplast which is largest among all and colorless and rod shape with circular also and found in underground part. Now function is for storage and reserve food material. Now three type on the basis of nature of food store. We have amyloplast store starch like in rice, potato, wheat. Now proteinoplast also called aleuroplast that store protein, example is maize. Now ileoplast that is also called oligosomes store oil or fat. Example is Ricinus that is castor seed. means Ricinus is in dicot with endosperm. Dicot with endosperm. Now we have second, that is what? Chromoplast. Chromoplast is colored plastid that is other than green. That is other than green color. It is second largest plastid with to provide color to fruit, seed, and flower. So chloroplast, formed by chloroplast or leucoplast. So mainly chloroplast, tomato, chili, and leucoplast, what? Carrot. Now green tomato change to red due to chlorophyll pigment change by lycopene, and chili green chili to red due to chlorophyll pigment change to capsanthin. So green tomato, lycopene, green chili, capsanthin. Now other color plastids here, rhodoplast, that is red algae, that is red plastid, and pheoplastic, that is brown algae. So it is brown plastic. Now chloroplast. So chloroplast at last discovered by Sack and named by Simper. It is called what? Kitchen of plant cell or photosynthetic apparatus or autoplast. Now shape is here discoid, tablet, or biconcave. Now it is found in all photosynthetic organisms except photosynthetic bacteria and cyanobacteria. In photosynthetic bacteria and cyanobacteria found in what? Chromatophores. So photosynthetic bacteria and cyanobacteria alternative found, that is called what? Chromatophores, and that is stores starch temporarily. Now shape of chloroplast. So cup shape Chlamydomonas, spherical shape Cholera, spiral ribbon Scleriform spirogyra, gilded ring collider horse shape Eulothoryx, and reticulate net-like shape Oidogonium. Now continuing to our chloroplast, in the plastid type we are discussing what? Shape of chloroplast. So stellate or star shaped chloroplast found in Zygnema is like double star. You can see a star like a starfish. So like discoidal shaped in Boucheria chara, biconvex oval shaped in higher plant. Now here number of chloroplasts. So single chloroplast found in Cholera and Chlamydomonas. Single chloroplast in each cell chloroplast is found in Eulothoryx. Double chloroplast found in each cell is Zygnema. Number of chloroplast is maximum in mesophyll cell. So Spirogyra has 1 to 16 chloroplast cell. Chloroplast differences into grana and stroma. So grana is the what? Grana is the arrangement, and stroma is the like liquid. So grana is side for light reaction also. A grana. So grana chloroplast found in commonly in algae, and bundle sheath chloroplast found in seafood plant. Agonal chloroplast contain thylakoids which are not stacked by what? Laminal chloroplast. So laminal chloroplast is by the lamella. So lamella, that is on the lower plant. Now thylakoids. So thylakoids. Now in thylakoids we have what? Structure unit of chloroplast, and it is like this, this is called what? Thylakoids. And these two thylakoids is connected by the membrane that is called what? Photosynthetic lamella. You can see. Now this photosynthetic pigment this present in this thylakoid, that is called quantasomes, and the quantity sums is 230 chlorophyll, quantity sum contain 230 chlorophyll, 160 chlorophyll A and 70 chlorophyll B, also with also 50 carotenoids molecules column. Now quanta sum in bacteria contain only what 50 chlorophyll molecule. And unit of grana is also called baggy trouser. Grana interconnected by stroma lamella or feet. So like this is contained by here this like liquid stroma that is connected by stroma lamella. Grana interconnected by means of this is called what stroma lamella. This is called what stroma lamella. And grana interconnected by stroma lamella. So stroma is like ground substance, like liquid. Now this stroma contain matrix, DNA plus ribosomes, and matrix contain enzymes for direct reaction or CO2 fixation. With blue-green algae lack chloroplast site of photosynthesis. In this blue-green cyanobacteria is called photosynthetic lamella. So these two connected, that is called what? That is called stroma lamella, because connected these two grana is connected, so that is called stroma lamella. Now we come to nucleus. So actually nucleus is not cell component. We can notice in cytoplasm we can say that cytoplasmic organelles in one side and other side is what? Nucleus. So nucleus is not cytoplasmic part. It is what? Protoplasmic part. So in protoplasmic come cytoplasm and nucleus. So nucleus originally two layer, that is called double layer membrane. So discovered by R. Brown, also called brain of cell, and it is not organelle. I have said you. Now largest extra cytoplasm. So it also not containing cytoplasm. The cell, the smallest component of cell is microfilament, and hammering proved. So hammering proved what? That nucleus is control center of cell. And number of nucleus, you can say that uninucleate one nucleus, one cell is common, two nucleus in a cell in paramecium, and on the AB, that is Ascomycetes and Basidiomycetes of the fungi, multinucleate is of two types, that is syncyte, and one is what? Conocyte. So first syncyte is by the fusion of what cell? So like this is many cell that is fused to make one, that is syncyte. Example in bone marrow, skeletal muscles, and ascaris epidermis also contain this earthworm. Now cenocyte is what? Their nuclear division only within a cell, but no cytoplasmic division. Without here, without means no cytoplasmic division here occurs. Now come to this. So this is like within a cell the nucleus only divide, no cytoplasm divide. Example of this conocyte means where nucleus only divide and no cytoplasm division. So the example should be hyphae of Phycomycetes, latex cell, Xanthophysi, Vaucheria, and Botrydom, with unnucleated cells without nucleus. And in plant, which what cell contain no nucleus, that is sieve tube at maturity, because sieve tube gain by the help of what? Companion cell in the phloem. We will study in that mammalian RBC we have studied. Now prokaryotic incipient nucleus means no true nucleus is present. Now structure of nucleus contain these four parts, and this chromatin also contain what? 4 parts, and we will study. The first part is nuclear membrane. Nuclear membrane is the outer membrane, and this membrane is also what, two layered because of nucleus is dimembrane or two membranes layer. So it contains double layer, and this double layer contains lipoproteins. There is also a space that is called perinuclear space, and it lack ribosomes because inner component of it is present, and it's separate from nucleoplasm from cytoplasm. So cytoplasm is where the cell organelles type of present, and that nucleoplasm is inside nucleus. So this is the boundary. And here also pore that is called octagonal, and gate that is called annulus. This is the nuclear pore and gate. Now second part of nucleus is nucleoplasm. Nucleoplasm is that liquid that is called nucleoplasm, where mainly no viscosity, where cyclosis is absent due to viscosity is what, constant, and it's include what, nucleotides here nucleotides. What, nucleotide is also what, nucleotide is also held. Now other third part is nucleus. So nucleolus is like one baggage part of the some molecule which contain ribosome, and that is factory of cell, and it is the largest part of nucleus also because of the bundles contain what, rRNA and but no DNA. So rRNA, so ribosome factory of the cell. And here second constriction, by the process of second constriction, biogenesis means what, formation from the second constriction. So second constriction is what, satellite chromosomes. So from the second constriction of satellite chromosomes, this nucleolus is formed. Now part of nucleolus. So now nucleus also has what, four part. So nucleus also four part, that is amorphous matrix. So amorphous matrix contain RNA, non-histone, acidic protein and calcium. So remember only granule part. So granule part contain ribonucleoprotein, and fibular part, that is contain RNA and protein. Precursor of what, granule. So this form what, granule. Now we have nuclear chromatin, from that nucleolus. So this contain rRNA. So due to rRNA it has what, ribosome factory of cell. Now chromatin, this is the fourth part. This is the fourth part and main part. Here this term given by Fleming, named chromosomes given by Weyers, and made of DNA which is like acidic and negative charge, and protein, histone protein, which is basic but what, positively charged. So this is what, basic and positively charged, that is histone protein. So nucleosomes here occurs. So here in nucleosomes 8 histone molecules given, of that is of 4 type, and the core body made up of what, histone protein, known as nu body. Now two nucleosomes joined by what, linker DNA. So linker DNA is for two nucleosomes joined. So in chromatin two linkers DNA. No, in chromatin we have a condensed form that is called chromosomes. So it got stained by Flaggen stain, and chromatin, that mass is called chromatin mass is called nucleosome. So these two nucleosomes is joined by linker DNA. Now chromatin contain two part on the strain, that is light strain and dark strain. So light strain is activated and is euchromatin. Dark strain is heterochromatin and inactivated gene. This is also secreted in S phase. S phase in the life cycle of the meiosis. Now chromosomes is that is called physical basis of inheritance, DNA chemical basis of inheritance, and chromosome. Now chromosomes is what filamentous structure where genes carry and is the vehicles of heredity. The chromosomal theory of inheritance is given by Sutton and Boveri, and morphology of chromosomes is called karyotypes. So morphology is called what? Type, karyotype. And diagrammatic representation chromosomes is called ideogram. Haploid chromosomes is called genome. So like genome is a set of chromosomes, whole set of chromosomes that define all the genetic material. Now difference staining, that is heterozygosis, and saliva test Olympic gamine bar body, that is X chromosomes. Now ultra structure of chromosomes, if you see in the eukaryotic, that contain pellicles mean cover of that chromosome. Now matrix is ground substance, liquid chromonemata. So chromonemata is the thread, like this thread, and this is thread, this is thread makes what? This is thread makes that chromosomes. And primary constriction. So primary constriction is centromere, where two chromatids is attached. So like this is two chromatids. So this is in conjunction, there is this two chromatids is what? Attached. So two chromatids is attached. These two chromatids attached, that is on the point side with kinetochore, where a spindle attachment is also present. Now second constriction is called telophase and contain rRNA. Now secondary constriction occur, that is called satellite chromosomes, and telomere is the terminal end or seal end of chromosomes. Now type of chromosomes on the basis of centromere. So like this is like chromosomes, and here in between this is the what? Centromere. So this centromere contain what? Centromere, a terminal telocentric. This is anaphase. In anaphase this stage occurs. It is like T-shaped. Now acrocentric is subterminal on the top. This is also anaphasic phase and is also T-shaped, anaphasic. So acrocentric and telocentric is for anaphasic phase and T-shaped. Now submetacentric. So submetacentric is L shape, and this is also what? Anaphasic phase, and this is L shape. Now metacentric is for middle, and this is also anaphasic stage, and where V shaped is located. So this is for V shape. Mainly this is metacentric, and metacentric is in last V shaped. Now acentric means without centromere. Holocentric means whole surface is centromere. Example is Luzula. Now shape of chromosomes best study in what? Anaphase. So we have all that is two acro and telocentric. These two acro and telocentric T-shaped. This metaphase is L-shaped. And submetacentric is L-shaped and metacentric is V-shaped. This all in anaphase. So best study in anaphase and metaphase. Now here ratio length that is called centromeric index. Now we have other from cell component that is ER, which is first observed by Porter. Now it's developed in pancreatic digestive system, poorly in sperm. Its unit membrane is bounded by, now here this is only unit membrane, means single layered membrane. Now it is absent in prokaryotics and some eukaryotes like membrane, RBC, egg, embryonic and monocyte of WBC. With ER poorly developed in rapid dividing cell, example is mesometric cell. So no ER present in mesometric cell. Continuing to ER, so mainly the name is endoplasmic reticulum ER. So endo means in the inner side. So in the inner side, this contains three parts, that is cisternae, that is bag type, then tubules, then vesicles. And cisternae is not like in parallel, that is present in Golgi body. So this is like what, bag shaped. Now ER is also continuing from the nuclear membrane to cell membrane, that is what, RER. And ER continuing from one cell to another by the plasmodesmata, means connector, plasmodesmata, and that connector is called desmotubules. Now ER is called working bench. So working bench of the cell or endoskeleton. The name is endoplasm. So that is inner skeleton. So that is called endoskeleton and forms more membrane than 50%, and it contains 10% of the total volume of the cell. Now it contains two types, that is RER and SER. So this is rough endoplasmic reticulum, and yes for a smooth endoplasmic reticulum. So this is 2/3 and this is 1/3. Here 70S ribosome, here no ribosome found. So here ribosome found. So here this will attach in nuclear membrane, and here arise from what, RER. So this come from this. So from RER SER comes, and function is protein synthesis, glycosylation, and this function is lipid metabolism. So mainly main function is RER, that is attached from the nuclear membrane of the nucleus and contain ribosomes and forms what, protein synthesis and also SER. Broken RER, that is main RER, that is called microsomes, and part from RER, that is called ergastoplasm. Now nasal granules is the combination of RER, that is called nasal granules, and RER without ribosomes called transitional ER. SER is for myelinoid tissue. SER for the lipid metabolism. So myelinoid tissue is of the most photoreceptor in the eye retina. Now a special point is that sarcoplasmic reticulum in the muscle cell of the skeletal muscles. Here calcium help in contraction and fusion of vesicles, and microtubules spherocytes formed by SER microbodies. So microbodies that is formed from SER, and from the RER Golgi complex occur to give lysosomes. So how lysosomes come? Lysosomes comes from RER with Golgi complex. Now function of SER. So SER is for lipids. So in the liver cell, that is for detoxification and formation of glycogen and glycogenolysis, for the breakdown of glycogen into glucose. Now ER function like same endoskeleton of the cell and form pigeons of microtubules. Structure cytoskeleton helps in mechanical support due to a skeleton in the inner side. keep cell organelle in the position. Membrane factory, it can form what? Lysosomes like organelle. Now formation of vacuoles, lysosomes, gliosomes, Golgi bodies. Example. So Golgi body also formed from what? ER. Detoxification, you can say detoxification on the liver, and major component here phospholipid, that is inside lecithin. So lecithin is associated with the food. Now Golgi complex or lipochondrin, discovered by Golgi, found in nerve cell of the cat and wool, here first discovered by the Camillo Golgi. Now called iodosomes, deltoid complex strained by Becker's body, and that is called dictyosomes because only found in what? Plant cell. Now absent in prokaryotics and some eukaryotics like RBC, sieve tube, bryophyte, pteridophytes and fungi. Now here formation. Now this is formed by E endoplasmic reticulum. So here main is SCR. So SCR will form this, and here cisternae is parallelly arranged as we have talked, and since cisternae continue fusion that makes forming shape with two convex on the outer. And Golgi complex is called what? Golgi complex is called director or traffic in cell. This is the other name. Microtubules here never present, and Golgi complex will involve in zymogen granules, means formation of zymogen granules for the enzymes or what? Hormone synthesis, enzyme synthesis mainly. Now function is for secretion and storage, modified transport, and cell plate formation. With Golgi body's vesicles, we will form what? We will form middle lamella. We have studied of vesicles, spindle protein as called phragmoplast, help in formation of lysosomes. So this help in formation of lysosomes. Golgi bodies with ER, and formation of acrosomes in sperm mucilage helps in what? In root lubrication, means for the smoothness. Formation of yolk, glycosidation. What glycosidation is? Glycolysis will occur for making glycoprotein, glycolipid. So this is also important. Now we have a special point, enzyme ER and GB. So this main enzyme RER is for peptidyl transferase, and glycosyl transferase is for main enzyme in the GB. Now ribosomes discovered by Palade, so-called Palade particles, and ribosome is called engine of cell or protein factory of cell. Now a ribosome is smallest, and this is organelles combined with what? Lysosomes. So smallest cell organelle is lysosomes, but this is also smallest, and found in cytoplasmic organelles. So this is on the cell living component, ribosomes occur. Now continuing to ribosome. So ribosome is called what? Protein factory of the cell, and here in ribosomes most abundant in what? In cytoplasmic is present, and this is called what? Protein factory. Now this is made up of two, that is rRNA and protein. So rRNA from nucleolus. So ribosome formed inside what? Nucleolus, and protein, that is synthesis from cytoplasm. Now is called what? Ribosome factory of the cell is nucleolus. So and ribosome is itself protein factory. Now ribosomes also seen from cell organelles by fractionalization under ultracentrifugation, and ribosomes are in protein synthesis which is found in both eukaryotics and prokaryotics, except this RBC and sperm. Now, two type of ribosomes, that is 70S and 80S. So 70S in prokaryotics and eukaryotic both. So mostly prokaryotic in all, but in eukaryotic only in these two, because these two is semi-autonomous structure present. Now 70 years has subunit, that is 30 years and 50 years. But 30 years single RNA, rRNA, and 50 years is what? 2 rRNA. Now this 70 years RNA contain three. This is two RNA, 2 RNA that is single. 2 RNA that is single and 1 is single. So 3 RNA and protein is 55 years. So 55 years ribosomes in mammals, 55 years ribosomes in mammal mitochondria, and not 70 years. Now 80 years ribosomes that is found in eukaryotics only, not prokaryotics. Now in free stage and in RER. So RER is for protein secretion, and subunit here is 40 and 60 years. So 60 years has 3 RNA and 1 single. So total 4 rRNA and 80 protein. So 2 ribosome connected by magnesium ion, which is formed by the dimer and attachment. This attached to mRNA and called polysome. Now during protein synthesis, this is attached by mRNA and forms what? Forms cell organelles, that is protein, and this also including plastic mitochondria and nucleus inside it. So these two semi-autonomous, so 70 years. Now here the question given, whichever flowing is electronic microscope. So this ribosome, because of very small. Now other is our microtubules. So this microtubules is also single membrane, but ribosome was what? No membrane, membraneless. Now this take place in oxidation reaction, then other than respiration. So here this microbody include lysosomes and spherosomes, two types. So first, that is lysosomes, that is polymorphic organelles. So this is a structure from neurospora, absent in all prokaryotes, present only in eukaryotics. So lysosomes is mainly on the animal and eukaryotics. This is also found in some plant like neurospora. So first that is extracted. Now cotton seed and G maize means maize, and found in WBC, acrosome spleen for the adaptation. So mainly lysosomes is what? Killing. So this is killing cell, and that is called acid medium. It activates in acid medium with 50 type of hydrolytic enzymes. So this is main line, that is called acid hydrolysis. So acid phosphate makers of lysosomes and protease contain hydrogenase, capsaicin and nuclease with DNA and RNA, nuclease, endonuclease, exonuclease, endo and exo. So here 50 type of hydrolytic enzymes found in acidic medium. So this activate only in acidic medium, and this is for the cutting. So restriction endonuclease, this is for cutting, this cuts, and lysosomes for enzymes. So mainly lysosome is for what? Making date burn cell macrophage. So lysosomes polymorphism 1 degree, so newly formed, that is Golgi body, and secondary lysosomes formed by the primary and food vehicles, residual body with undigested food material, and autophagic, that is digest cell organelles. So this digest cell organelles is in autophagic lysosome. Now this main function is intracellular formation and digestion of macromolecules. So for self-destruction, sputum second type found in only eukaryotic plants. So lysosomes only on animals and sputosomes only on plant. Now this is for oil storing. So called as oil storing bodies and present on endosperm of oil seed. Now here also hydrolytic enzymes, like there what hydrolytic enzymes occur, hydrolytic enzymes. So here also hydrolytic enzymes in plant and helps in synthesis and storage of what fat. So lipogenesis developed, this is developed from ER. We have studied previously, so that lysosomes is formed by GB and ER and disappeared after germination, biosynthesis of cutin and wax. Now third type is peroxisome. This is like not much important because peroxisomes deals with both plant and animals and used in photorespiration. You can see now some I think we have 4 or 5 types of microbodies. So we'll start. Continuing to peroxisome, that we have told that both in plant and animals it is for what breakdown enzymes and what bioluminescent, and here main function is photorespiration in this three, that is mitochondria, peroxisome and chloroplast. So photorespiration occurs in 3 organelles, that is chloroplast, peroxisome and mitochondria. Now beta oxidation here for fatty acid conversion into acetyl CoA. Now fourth type of microbodies, that is glyoxysomes, found in plant and fungi. So this is all largest, and here common example neurospora. This is for what, this was beta oxidation, this is also for beta oxidation in the Krebs cycle fat. So here fat broke down in the glucogenesis. Now lomasomes present in fungi only, in the fungal plant only, hyphae. This is for what cell wall formation, cell wall lysosome. Now transosome, this is what three membrane layer or trimembrane, given trimembrane layer found in ovarian follicles for the synthesis of yolk, or in other animal vitellogenesis, means formation of yolk with the help of Golgi birds. But on the ovarian follicles of the bird only we have transosomes. Now vacuoles in plant and only in plant cell only, not in animal cell. Now this is a storehouse of the cell, that is called what, tonoplast, also because of single layer and living. Now fluid present inside, that is called cell sap, non-living, and is acidic or neutral, and the reserve material is insulin here. Now vacuoles contain pigment anthocyanin. So anthocyanin is for color, that is red, blue, purple. Now anthocyanin pigment called as what, vegetable chameleon, means change color due to environment, like acidic red in basic blue or purple. Now the anthocyanin is for what, a strong sunlight, shield also. So red rose is red due to anthocyanin pigment, purple color due to anthocyanin pigment. So everybody anthocyanin pigment. And now here most evident element present, that is potassium. Now vacuoles here help in what, vacuoles help in osmoregulation. Now gas vacuoles in somewhat in watery plant, that is the buoyancy of the cell. Now cytoskeleton structures, this composed of 3. We have studied MT, MF and what, F, like this. So first, that is MT. So microtubules that is hollow, made of tubulin protein, and 13 pro 13 protofibers. Now this is found in centrioles, cilia, flagella, spindle fiber, basal body. Now microfilament. So microfilament is elongated, cylindrical, solid, from protein actin, helping cell plate formation. Microfilament. Intermediate filament is made of keratin, helping what nucleus to maintain position. So intermediate filament is for position maintenance. Now centrosome with centrioles. So centrioles is only found in what? Eukaryotic. So absent in this prokaryotic and angiosperm. In animal only, and that is ciliated algae. Now these centrioles are hollow, made up of microtubules. Now these both centrioles and centrosphere make what? Centrosome. Centrioles and centrosphere make centrosomes. And centrioles give wheel appearance, cartwheel appearance. Replication of centrioles on S phase, division on prophase. Now this attach on the basal's body with 9+0 attachment, and important is cell division. Centrioles associated with cell division. Now two type of cilia and flagella. This is for what? Locomotion type. This is numerous and this is few number, shorter and longer. It is vibratory and it is non-vibratory. Now here given after cilia and flagella in prokaryotic how this happens. So in prokaryotic this is 9+2 arrangement is absent, and this is not made of microtubules and tubulin protein, and it contains single fibril, that is called monofibril, and flagellin protein with unmembranous. Now in eukaryotic it is double membranous, and 9+2 arrangement is present with 11. 9+2 is 11, that is microfibril. Now we have cell inclusion or eugastic substance. So cell inclusion is like non-living substance of the cell. I have told you in the third class. Now this, due to the activity of protoplasm, there is non-living metabolic substance, inactive substance is deposited on the cell inclusions. Now this is like three types. First is reserve storage material like carbohydrates, protein and lipid. This is what? For the reserve, for carrying what? Energy purpose in the lack of food. So carbohydrate is most abundant monosaccharide, most sweetest is fructose, and this fructose is present in semen and honey. Now sugar is nectar, honey, sugar. This is also fructose, but grapes is on the glucose. Galactose is nerve sugar. Insulin in dahlia, so dahlia starch. Now alluron grains in cereals. Cereals means for the maize or rice. Largest starch grain in grana, smallest in rice. Now lipid is like fatty acid, unsaturated fatty acid. Example, oleic acid and linoleic acid, and which is amphipathic, means both hydrophobic and hydrophobic in nature. This is like waxes belongs to here. Now protein by the activity of amino acid combination, protein form. Smallest amino acid is glycine, complex is tryptophan, and first discovered is asparagine with sulfur containing these three, CCM, and amphoteric. This is also amphoteric. Most abundant protein is rubisco. Rubisco is for what? Plant. Smallest protein is ACTH. ACTH on the what? On the hypothalamus gland or pituitary gland secretion. Now gelatin in maize, originating in rice. Now secretory product, we have now secretory products. So secretory products contain what? Enzymes. So enzymes termed by coenzyme, and here haloenzymes. So here in active site attached during what reaction. Now nectar is mixture of glucose, sucrose and fructose. Now this attracted insect for pollination. Now here pigment is for yellow pigment xanthophyll and orange color carotene. Now pigment anthocyanin, purple for the fruit. Secretory product. Now third type is secretory product like alkaloids. Alkaloids means here nitrogen containing substance in the waste. This is for the protein waste in alkaloids. This is insoluble in water and also used in medicine. Now atropine, example atropine in the eye drop, Atropa belladonna. Quinine in the malaria used on the bark of concane. Morphine from the poppy plant, this is opioid type. Nicotine, this is also drugs type. Rasparin, this is for the snake bite. Ephedrine for the blood pressure on the gymnosperm. Ergotin for the fungi, and caffeine, coffee, vaccine, colchicine. Colchicine, that is called mitotic poison, means no mitotic will be formed due to spindle fiber dissociation, means disturb or dissolution. Now quinine reserve, and this is for treatment of high blood pressure. I have told you this and high blood pressure. And atropine is for heart disease, or you can say eye dilution. Now second is tannins, that degradation product of carbohydrates, bitter in taste. It is in the highest in young fruit. Now we have C number type, that is latex, which is yellow or white fluid suspension for the protein gums. Now main source is for rubber production. Now this is for the rubber. Example is Hevia versalis, Ficus elastica, Indian rubber. Papain is for protein digestion. Now we have glycoside is for the carbohydrate degradation product. Example is saponin. Next is gums for the cell wall dissociation, that is gummosis essential oil. Example in leaves of citrus. Resin for the essential oil also. Now like example is oleracin, Candida balsam, and gum resin, hing. Now organic acid, citric, malic, oxalic, tartaric acid. Mineral crystal, calcium carbonate, calcium oxalate, sorry. Now resphid and spheroids doses and aggregates, calcium carbonate like crystolith and crystal of silica in the grass. And chloroplast contains spore is in Funaria, Euchristium cell wall grass, and Euchristium with what? Silicon.